I think Dr. Bogota will be late, and we just send the reminder email to Dr. Bogota. And I think we can start on time, and we will try to contact again with Dr. Bogota.
Okay.
Hi, Professor Ramandari. We can't hear you.
Hello, everybody.
Hello.
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 Yes, we would like to introduce Professor Ramandari is the executive chief editor of our journal, and I think we can start.
Okay, now let's start a webinar. Dear distinguished participants, ladies and gentlemen, welcome you to the webinar, DIRT, Liver Diseases, Liver Cancer, Prevention from the Source. This is Ada Zhong, the editor of Heptama Research. It is a great honor and pleasure for me to express our sincere appreciation to all of you for being here. On behalf of our staff and the editorial board of the journal. In particular, we would like to say a big thank you to the webinar chair, Professor Ludovico Abanavoli. Thank you for having organized this big event, and to our guest speakers' strong support. We hope everyone will enjoy this academic webinar. Before we get started, let's welcome our marketer, Ms. Zoe Wang, to announce the webinar etiquette tips. Zoe, please.
Thank you, everyone. In order to maintain the order at the meeting, please follow the rules. Number 1, please mute your microphone during the whole meeting. Turn off your camera when speakers are talking. Do not make any distracting noises. Number 2, all participants are only allowed to ask questions in the discussion section. If you have important questions, please write down your question and try to wait for your turn during the allotted time. Or please send it to the chatting room and we will note it down. Number 3, please ask questions shortly, concisely, and slowly. Repeat your question if necessary in the discussion section. Number 4, please introduce yourself first before asking questions. Number 5, all registered participants will be offered the certificate. Anyone who did not register on the webinar and would like to have the certificate, please contact the editorial office after the webinar. Thanks a lot for everyone's cooperation.
Thank you, Zoe. Now let's warmly welcome Professor Ludovico Abanavoli to deliver a welcome speech. Welcome.
Distinguished colleague, speaker, everybody, thank you to be with us today in this international webinar organized by the journal Hepatome Research. I am grateful to the journal teams, in particular to Ada and to Victoria for his support in the realization of these events. Today we speak about non-alcoholic fatty liver disease. Non-alcoholic fatty liver disease is the most frequent chronic liver disease worldwide. It is estimated that in the world, about 25% of general population present this condition. Now we know that NAFLD is a multifactorial disease and involve many specialties and several factors like to insulin resistance, inflammation, cytokine imbalance, dysbiosis of the gut microbiota, change in innate immunity, and environmental and genetic factors. In the last 2 decades, many studies are published on this topic. The aim of this event, of this webinar, is to update the knowledge on NAFLD with regard to lifestyle, diet, treatment to manage this condition in clinical practice. Thank you one more time for your presence and your time with us. Good. For the first lecture, I invite Professor Vladimir Milivojevic from the University of Belgrade, Serbia, to present his presentation, MAFLD, Nutrition and Microbiota, Bidirectional Link. Please, Professor Milivojevic.
Thank you. Thank you, Ludovico, for a nice call. Thank you, Hepatoma Research, for this tremendous event. Thanks to Ada and Victoria for good organization. Thanks all speakers, and I hope so that we will perform that webinars frequently, because this is a very important topic. I choose this topic because it's maybe when we speak about MAFLD and NAFLD, it's for me is the most important discuss about microbiota and about nutrition, about diet. Next slide, please. I'm Vladimir Milivojevic from the Clinical Center of Serbia. Next. In the agenda, I will speak about paradigm shift, about new definition of MAFLD, diagnosis and theology, microbiota and microbiome composition and diversity, and the impact to the MAFLD and the liver, dysbiosis, which factors can trigger that, nutritional dysbiosis, and gut-liver axis in the key pathogenesis points. Also, I will mention some treatment options, new treatment options for treatment of MAFLD. As you know, we recently make a new name for NAFLD to MAFLD, and a lot of us here were included in like experts around more of 130 countries to reorganize name and to endorse changing of name to metabolic dysfunction associated fatty liver disease. For me, this is very important step, and this is a very appropriate new name for this entity. As you know, hepatic steatosis, when we find out and when we see that detect either by liver biopsy or imaging techniques or with non-invasive tests like blood biomarkers, steatosis. If we have obese or overweight patient, and if we have diabetes mellitus, patients with diabetes mellitus, we can immediately establish diagnosis of MAFLD. With lean and normal white patient, hepatic steatosis together with two of that presenting factors which were included in previous NAFLD diagnostic steps can be enough to establish diagnosis. What is the better performance of MAFLD in relation with NAFLD? Because we with MAFLD we can have also a dual etiology of fatty liver disease. We can see autoimmunity like etiology. We can see hepatitis like etiology. We can see another factors which are included, which can be in relation with our diagnosis in our patient. also for me is MAFLD, metabolic associated fatty liver disease, is more. It's more. Hello, can do you hear me?
Yes.
Yeah, sorry. Next slide, please. I forgot that. Yes. Sorry, I had a problem. Yes, next, please. Yes. And what is complex etiology of progression of and establishing of metabolic associated fatty liver disease? For sure, triggers and bad factors of etiology are diet, microbiota, environmental stress, and genetics accessibility. And nowadays hello, it's everything okay?
Everything is okay. Everything is okay, Vlad.
Just need to come back my presentation. No, yes, okay, nice slide. Okay, thank you. Sorry, no, next slide, please. That etiology can lead to progression of steatosis and fibrosis. Fibrosis is point of no return. And for us, for clinicians, it's very important to recognize that problem on the beginning and to manage this problem and to make some possibilities to prevent cirrhosis and hepatocellular carcinoma. Nowadays, we can see that with westernization of diet and with more prevalence of obesity, we can find a very, very important factor of liver cirrhosis from the steatosis and from MAFLD. It's a very important thing is to targeting that group of patients and to possible to cure that patients on the beginning and in the good phase. Next slide, please. And what is next slide? Hello. Thank you. It's very important to distinguish terms microbiomes and microbiota. Microbiota are the all the organisms, microorganisms, which is included in our body, in the gut, and the bacteria, protists, algae, fungi, virus. Microbiomes is genetic materials which is produced, which is from that microorganisms. And like Hippocrates said, all diseases begin in the gut. Maybe nowadays we are the closest to say that is very, very big too. And as you know, gut microbiota compositions varies depending of part of our next slide. Depends of part of our part of gut. And in the stomach, we have a little bit sterile environment with lactobacillus, streptococci, some candida, and in some cases Helicobacter pylori. But small intestine and large intestine are richful and with bigger diversity and with much more species, which is lactobacillus, coliform bacteria, streptococci, bacteroides, bifidobacteria, fusobacteria, and various clostridias. When we are going from mouth to rectum, we are changing our pH gradient. Also, we are changing composition, and we have much more bacterial species across our gut. Next slide. And what make implication for our microbiota. Everything, I can say everything for our birth, pregnancy and infancy, and diet from our mother, diet, gestational diabetes, environmental exposures, antibiotics, drugs, things that we are breastfeeding or we are in artificial formulas of milk formulas, different pharmaceuticals, but age also, infection during our childhood, during the infancy, during the adolescence period, and many more factors which can establish good and potent and well-balanced microbiota, or can be with bad impact on our diversity on our microbiota. Next slide. Next slide, please. And what happens with dysbiosis in the context of Smallholder? Altered microbiome composition leads to low diversity. That can lead to smaller amount of Bacteroides, higher abundance of Firmicutes, Proteobacteria, and smaller amount of Bifidobacterium and Erythrosphaeria. Low abundance of some bacterial species and high abundance of another species lead to higher level of pro-inflammatory cytokines and leaky gut to higher permeability or intestinal membrane. We are going to establish gut-liver axis that all lead to damage of our liver with big amount of endotoxins, which can make changes in structure of our liver. Next slide. And next slide, please. And Mafalada and dysbiosis have to slide before, please. Slide before. Yes, Mafalada and dysbiosis shared triggers. You can see stress, smoking, recurrent infections, susceptibility, genetic susceptibility, aberrant immune system, and nutrition can be together factors which can trigger concomitant dysbiosis and Mafalada. If it's nutrition rich in animal proteins and deficient in dietary fiber, we can establish Mafalada. Also, same factors can manage from eubiosis to dysbiosis establishment, and that two factors are like my title of my presentation said, bidirectional link between liver and gut, between dysbiosis and Mafalada. Next slide, please. And what is make that diversity and that make microbiome fluidity? Composition and diversity of gut microbiome begins to form as early as during intrauterine period. Diversity is largest in early years of life, but somewhere and in somehow we have smaller amount of good bacteria like a Bifidobacter, Lactobacillus bacteroides, and higher abundance of Horsidicea, Staphylococcus, Enterobacteria. Big studies revealed that dominant influence of extrinsic factors like as nutrition is comparable and is most effective than genetic factors. We now know that microbiome manipulation during the childhood can permanently change microbiota diversity and and abundance of good bacteria. Next slide. And what is good manner and good nutrients for our gut? For sure, dietary fiber, lipids, and some parts of carbohydrates. Most important thing for nutrition and for our microbiota are prebiotics like butyrate and propionate, acetate, short chain fatty acids, which can be good nutrition for our good bacteria, for Lactobacillus bifidobacteria, and which can lead to better environment for our gut membrane. Next slide. And as you can see, we can be in good balance with healthy nutrition, and our liver will be healthy. If we have imbalance in nutrition, our liver start to be with steatosis. In further course, we establish fibrosis, even cirrhosis. What is a balanced nutrition for our gut and for our liver? For sure, that are natural probiotics, polyphenols, omega-3 acids, glutamine, carotenoids, oligosaccharides, fibers, and L-tryptophan. What is imbalanced nutrition which can lead to higher level of leaky gut and of dysbiosis? For sure, high saturated fats and high fructose. Next slide. And how nutrition is targeting our gut and our liver with glucotoxicity and lipotoxicity. Nutritional factor of sugars and animal proteins lead to obesity, to insulin resistance, to increase fatty acid uptake, to lipotoxicity, and to oxidative stress. On the other side, Mediterranean diet can lead to a smaller amount of energy, lead to smaller intake of dietary sugars and fats, higher vegetable proteins, smaller intake of salt, bigger intake of fiber and polyphenols. That can lead to prevention of inflammation, of insulin resistance, and that kind of diet can lead to insulin sensitivity. What is effects on the liver? Impaired glucoregulation leads to inflammation, to triglyceride and cholesterol synthesis, oxidative stress, and to synthesis of many pro-inflammatory cytokines and endotoxins, which can be very dangerous for our gut and our liver. Next slide. And what is about that by directional link of nutrition and dysbiosis? As you can see, regular diet is homeostasis, but we can, when we have Western style diet, we have gut inflammation and metabolic syndrome. High fat diet can be protective if we decrease intake of sugar and if we increase intake of omega-3 acids. Next slide. Next slide, please. Next, thank you. Nutrition can make local and systemic effects. As you can see, Western type of diet leads to microbial dysbiosis, mucus layer degradation, type 2 diabetes, obesity, chronic heart disease, cardiovascular disease, chronic pulmonary disease. Another side, fiber, indoles, and vitamins prevent obesity, prevent intestinal homeostasis, and intestinal immunity. As you can see, nutrition effects of microbiota composition can be various. Intestinal barrier function is the most important thing. We can regulate our intestinal barrier function and our gut immunity with very balanced and very healthy diet with high amount of fiber and with small amount of fructose and non-saturated fatty acids and animal proteins. Short-term nutritional alteration can transitory change gut microbiome, but long-term nutritional alteration significantly change in gut microbiota. We need to insist to our patients to be with long-term nutritional alteration, to be to reduce constant in the effort to long-term alter nutrition habits. Next slide. Next, please. Next. And what happens in gut-liver axis? Gut microbiota dysbiosis. No, previous slide, please. Gut microbiota, sorry, previous. Yes, gut-liver axis dysregulation. Gut microbiota leads to dysbiosis, bacterial overgrowth, decreasing of viral diversity, and decreasing of butyrate. That can lead increased permeability of mucus layer, epithelial barrier dysfunction, gut vascular barrier dysfunction, gut-associated lymphoid tissue pro-inflammation activity, and that all can lead to endotoxemia and to increase activation of pro-inflammatory pathways, endotoxin-dependent activation of hepatic Kupffer cells, increase inflammation of CD4, CD8 T cells, and macrophages. That can lead to steatosis and fibrosis of the liver. Next slide, please. What is leaky gut? Leaky gut is the bigger permeability of our gut membrane and various endotoxins which can go in systemic circulation, which can lead to inflammation, fatty acid oxidation, intestinal permeability, and another, together with epigenetic and environmental factors and genetic susceptibility, can lead to serious danger of our liver. Next slide. And what we can say about short-chain fatty acids? They have a controversial role in NAFLD and NASH.
Unmute your phone, please.
Yes, can you hear me? It's okay.
Yes, it's okay.
Yes, we
Lars,
can.
It's okay.
That systemic butyrate can lead to insulin sensitivity. Next slide, please. And butyrate is also good nutrition for our bacteria in our gut. Also, it's a good protective factor for enterocytes and colocytes. And the Firmicutes bacteroidetes ratio infrequently cited in the scientific literature as a hallmark of and biomarker of dysbiosis. It's very important with butyrat. We can make a very high ratio of that type of bacteria, and we with butyrat intake we can prevent dysbiosis. Next slide. Altered bile acid metabolism. Next slide, please. Altered bile acid metabolism is very important things also. TX receptors, it's in lipid homeostasis, also in bile acid homeostasis. And new drugs now, which I will mention later, are target for FX obeticholic acid, and new drugs which in phase 3 of study can be a very useful agent to improve lipid metabolism and glucose metabolism and bile acid metabolism, which is very important in the pathogenesis of the dysbiosis and leading of MAFLD. Next slide, please. Next, please. Thank you. Bile acids changes as markers of present and severity of NAFLD and MAFLD. As you can see, it's very important and significantly changes of bile acids in presence of NAFLD and NASH, and that changes are more exaggerated when we have severe NAFLD and MAFLD. Next slide, please. Next. Next, please. Hello? Yes.
Yes, Vlad, yes, we hear you.
Yes, small. Yeah, I spoke about dysbiosis in the large intestine, but it's very important small intestinal bacterial overgrowth and toll-like receptor signaling the patients with no alcoholic fatty liver disease. That we need to speak about small intestine. Small intestine is very important for pathogenesis of NAFLD, MAFLD, and the bacterial overgrowth in small intestine leads to endotoxemia also and leaky gut and liver injury. Next slide. And what can be next, please? Liver. What can be treatment targets for our patients? Bile acid, dietary fat, bacterial products, and we have a lot of studies and a lot of suggestions to establish combination therapy to different drugs to establish good goals for our patients. GLP-1 agonist, FX receptor agonists have promising results. Also, next slide. Mediterranean diet is very important part of that treatment procedure. As you can see, EASL recommendation, clinical practice guideline for the management of non-alcoholic fatty liver disease suggests weight loss reduction up to 10% with Mediterranean diet by energy deficit of 500 calories per day, low fat, low carbs, Mediterranean diet, dietary composition modification, reduced fructose, and reduce with high fibers, high fish, high vegetables, low cholesterol, low sugar, with coffee drinking 2 or 3 cups per day without alcohol consumption. Mediterranean diet is the only approved treatment choice for the NAFLD and MAFLD. Next slide. And for me, is dilemma: do we need to back to the beginning and be like current clinical trials said, NAFLD patients with one therapy to establish some effects, or in a group of patients we will be without effects or with adverse effects. I think that is very important thing to be with precision therapy, tailored therapy. We need to see novel clinical trials about more personalized therapeutics, which will firstly make stratification of our patients with tailored based on the MAFLD patients' heterogeneity, microbiota phenotype, metabolic phenotype, and gene susceptibility. Establishing that kind of therapy can be good and promising for next period and for our success with that patients. Next slide. And finally, I will conclude with some points that MAFLD contributes to raising patients' awareness of NASH. It makes the diagnosis easier, lifting of the NAFLD-related stigma, building networks with other metabolic disease with a high risk of NASH. Microbiota dysbiosis, decreased diversity and gut-liver axis dysregulation are present in patients with MAFLD, may contribute to patient stratification and effective therapy options. Our therapy may contribute to patients' stratification and effective therapy options. Short-term diet changes lead to transitory microbiome perturbation. We need to give our patient long-term diet changes that can lead to less inflammation, to less endotoxemia, to preventing of SIBO, leaky gut, consequently to preventing of liver injury. High MAFLD phenotype heterogeneity, microbiota phenotype heterogeneity at complex gut-liver nutrition interrelationship warrant for a precise tailored therapy aimed at different key points of MAFLD pathogenesis. Thank you for your attention.
Thank you, Professor Milivojevic. Thank you too for your presence from Dubai. You are in this moment in Dubai. I know that
Yes.
your flight on. I wanted to ask you briefly only one question, because after you come back to your country, what do you think about this new definition of MAFLD? This new definition of MAFLD made by thousands of stakeholders worldwide is better or not of MAFLD definition?
Yes, we thanks to Professor Faut also we established new definition. For me, it's very, very better. It's very important that changing of the name of MAFLD to MAFLD, because this is exactly this name, metabolic associated fatty liver disease. It's a real definition of what happens with our liver and with our patients in that kind of diagnosis. In next time, I will in contact with Professor Faut. We will establish from Serbia's endorsement letter. We wrote that letter, and I need to just send somewhere to establish our endorsement to new definition of MAFLD from our Serbian Society of Hepatology and Serbian other society, which I'm coordinator, which is part of our project Dilemmas and Controversies in Medicine, which you know, and which you were part of that congress last month in the Montenegro.
Thank you.
Thank you so much. Thank you. Unfortunately, I have to go. I wish you good work in the further course webinar, and see you.
Thank
Goodbye.
you. Thank you.
Goodbye.
Goodbye. Goodbye, Vladimir. Thank you. Thank you, Professor Milivojevic. Now we continue the webinar. It's a pleasure for me to introduce a colleague, a friend, a very good friend, Professor Luigi Boccuto from Clemson University of South Carolina, USA. And the topic of his speech is genetic factors in AFOD. Please, Luigi. I remember you that you have 20 minutes, please.
Yes. Yes. Well, thank you very much for that kind introduction. It's a pleasure for me to be here. I don't know if I'll share the slides or someone else will share them for me. I'm good either way. I'll try to. Okay. Can you see the slides?
Yes. Yes.
Okay. Perfect. Okay. So, this is a very interesting topic, and it's a very complex one. It's complex because the phenotype, the clinical manifestation of this condition is extremely variegated. And you can see here already from the cover slide how we deal with different stages of this disorder. But as we will see soon enough, the complexity goes on with the clinical characterization and diagnosis of this condition. I will not spend too much time on the clinical features because other more renowned and more expert speakers will cover that. But I just want to focus on the pathogenicity and the mechanisms that are underlying the onset and then progression of non-alcoholic fatty liver disease. As you can see, the clinical definition is based, of course, on the features that can be assessed. So we're talking about clinical assessment, imaging, laboratory test, and of course, biopsy. But we do have different categories that are interrelated. So there is a progressive dimension that we need to consider. There is increased severity of the manifestation, and there is an increased decline of the and degradation of the liver functions. If we focus on the different subgroups of NAFLD, we can see that this complexity is even exacerbated by the presence of comorbidities. In fact, the alcoholic fatty liver disease is often present as well, but also other conditions that really open the doors to different pathogenic mechanisms involving metabolic disruption, immune imbalance, toxicity from diet and other environmental factors. So if we want to have a really encompassing approach to the characterization of the causative mechanisms underlying this condition, we must take into consideration the complex nature of this disorder. And if we focus on the dynamics of the onset of this disorder, we can see that they are closely interrelated with other organs. In fact, if we narrow down the pathogenicity of NAFLD, we can see that there are influences from organs around it. So first of all, the effect of the diet, but the interaction with the adipose tissue, the pancreas, the intestine, as was mentioned before, the leaky guts, and all the interaction between bacteria, the immune system, and then the ultimate damage on the hepatocytes. But that's not the only direction of this interaction, because there is an interaction from the liver to the other organs. And that is important because we have to contextualize NAFLD in the wider spectrum of the metabolic disorder. As we saw before, there is a series of comorbidities of disorders that can be detected in individuals suffering with NAFLD. And this is a summary of the possible pathogenic interactions, where we have disruption of the lipid metabolism started from the liver and then spreading towards mostly two main mechanisms. The generation of insulin resistance that starts in the liver and then spreads to the other organs, and the induction of inflammation. The critical point about the inflammation is the chronic feature of it. So it's not just an acute event, but it's something that once is established, it remains and affects not only the liver tissue, but several other organs. So this is quite important to understand the systemic consequences of NAFLD. And since we're talking about a complex disorder, it's good to take a step back before we approach the genetic factors involved in these complex disorders. The definition of a complex disease is the interaction of environmental inputs and the metabolic response based on the molecular and genetic predisposition of each individual. The characteristics in terms of pathology of these disorders are that they do not have a Mendelian mode of inheritance. So even if there are genetic features that may be very relevant, as we will see in a couple of slides, the segregation in the families, the pattern of inheritance, the distribution in the population, they do not follow the Mendelian pattern, which makes it more complicated for systemic assessment during, for example, counseling for the family or large population screening. The other point is that even if there is a genetic component, is quite often not monogenic. So there are several genes involved that contribute to the overall phenotype. And sometimes the quantitative assessment of the contribution of each gene is quite complicated because the threshold determined in the interpretation of the pathogenic effects of genetic variants are designed to assess the contribution of single variants. But in this case, we might have a network of multiple genes, maybe even multiple pathways that concur to determine the onset and progression of the liver disease. And then the third element of complex disease is the environmental factors. The environmental factors may play a role that may vary from simple trigger when the genetic component is relatively strong to the sole causative effect. So again, it's very complicated to assess what is the actual contribution of these environmental factors because they may vary from individual to individual. So once we have an overview of a complex disease, particularly related to NAFLD, what is the role of genetics? And what is the role of genetics now in the post-genomic era? Researchers consider the post-genomic era this period of time after the final results of the Human Genome Project have been shared, after the next generation sequencing, the large sequencing approaches have become more available in terms of cost, in terms of logistics. And so they open the doors to the world of omics, where the omics are really the ones that are targeting the DNA, so the genomics, as you can see here, the RNA, the transcriptomics, that started with the messenger RNA, but really now is extended to all types of RNA, proteomics, proteins, interactomics, so the interaction of multiple proteins, and metabolomics. So as you can see here from this cartoon, these approaches are aiming to assess at the vast level, the very broad level, the possible alterations in our pathways, in our metabolism, and they are targeting the DNA, the RNA, proteins, and the interaction of proteins, and then finally the metabolomics with all the metabolites of the product of these proteins. So this will give us really a very encompassing approach that will help us understanding the link between genetic factors and the final clinical presentation. And this is possible through the functional genomics. So an approach that is focused on isolating individual alterations or single alterations in the gene sequencing or non-coding sequencing, and then through high throughput technologies, extending this analysis, this investigation to the whole genome, collecting data and interpreting data through bioinformatic analysis. So really introducing that multi-layer dimension where we can connect different alterations, and then analyzing, interpreting the results. And I would like to add another step to this graphic, which will be the translational step. So applying the information collected from this functional genomic approach to the clinical practice. So really bringing the information from the lab to the bedside of the patient. And so in order to do this, today I want to speak about the connection between genes and liver. And I like to use an example to summarize three main categories. We can use an example for genes where we can have the good, that is HSD17B13, the bad, PNPLA3, and the ugly, which is CIP2D6. Why did I pick these? Of course, there is a citation, the famous Italian movie, but we will see how these genes really represent a paradigm of different potential effects of gene variation on the liver metabolism. We start with the good, which is this gene 17 beta hydroxysteroid dehydrogenase 13 that encodes a liver-specific enzyme that targets steroids, pro-inflammatory lipid mediators, and retinol. So if you remember, when we had an overview about the pathogenic mechanisms underlying NAFLD, one of the critical steps was the chronic inflammation. So this is an enzyme that really works by expanding, amplifying, exacerbating the immune response and promoting inflammation at multiple levels. So loss of function variants, so inactivating variants in this gene, have been associated with protective effects against NAFLD. So that is why we label that the good. And this is important not only to stratify the general population for their overall risk of developing NAFLD, but also because it provides potential molecular targets for treatment. And this is the translational dimension we were mentioning before. So variants in this gene may prevent or delay the progression of NAFLD. So if you remember the steps from steatosis to non-alcoholic steatohepatitis, and then liver fibrosis, cirrhosis, and hepatocellular carcinoma, all that progression at every step of this progression may be either prevented or delayed, depending on the overall effect of these variants, by HSD17B13 variants. Even, and this is a recent study, even in individuals that are carrying the variants on the bad gene, the PNPLA3. So you can see how there are many actors at play here, many potential genetic factors playing, and some of them are playing against each other. So that is why the multi-gene network is important, because we need to assess the overall contribution of genetic factors. So overall, what we learn is that inhibiting the activity of this protein through RNA interference and small molecule agents may prevent NAFLD progression. Then we move to the bad, PNPLA3. This is a gene that encodes a patatin-like phospholipase domain containing 3 protein, also called adiponutrin, that is a very important intracellular membrane protein that is triggered after feeding and during insulin resistance and promotes lipogenesis. So remember, again, if we look back at the pathogenic mechanisms that promote onset and especially progression of this disease, insulin resistance was a very important step. Lipogenesis, so disruption or dysregulation of lipid accumulation is another important step. And the very interesting thing about this gene is that there is one variant, this isoleucine to methionine missense change affecting the amino acid residue 148, that not only is a prominent predisposing genetic factor for non-alcoholic fatty liver disease, is also a prominent predisposing factor for alcoholic liver disease, and to my knowledge today is the only genetic variant that is associated to both alcoholic and non-alcoholic forms at very high level. But it's relatively frequent in several populations. You can see that the estimate on the overall population is around 20-26%, but it may vary in different ethnicities. And in the highest points in Latin American ethnicities, it may even affect or may interest more than half of the population. So this is a very important issue for community medicine, because stratification of the risk in individuals from different ethnicities may be extremely important for the management and treatment of NAFLD. So here we can summarize the effects of this variant, where the wild type isoleucine amino acid in PNPLA3 148 position is responsible for a balanced transition of lipids through the membrane, and so it keeps the lipid droplets under a certain size. And instead, if we have the methionine, there is a promoted lipogenesis with the generation of macrovesicular steatosis. So there are these big vacuoles, this big accumulation of lipids in the hepatocytes. And then we move to the ugly. Now, why did I label this the ugly? Because it really creates a lot of damage, even if it doesn't seem to have a clear purpose. So like the character in the movie that is sort of messing around, going one direction or another, this one is not directly involved in NAFLD, but I wanted to pinpoint the role of this gene because it's extremely important in pharmacogenomics. And for a complex and systemic disorder such as NAFLD, the evaluation of the pharmacogenomic predisposition, the genetic background underlying interaction with drugs is critical, because as this condition affects multiple systems and multiple organs, therefore the management and treatment must take into account medicines, pharmaceutical compounds that will target multiple organs. And this is a gene that encodes a monooxygenase that localizes in the endoplasmic reticulum of hepatocytes and metabolizes, just this protein metabolizes about 1/4, about 25% of the commonly prescribed drugs. So it's part, of course, of the P450 family, the largest cytochrome family in the liver, but it's responsible for the way our body responds, interacts with 1/4 of the commonly prescribed drugs. And you can see here there are drugs from different categories and different families, from antidepressants to antiarrhythmics and analgesics. The thing is, this is an extremely variable gene in humans. It's highly polymorphic. There are multiple alleles recognized for their overall effect and defined as poor metabolizer alleles, rapid metabolizer, or ultra rapid metabolizer when you have 3 or more active copies of the genes due to the gene duplications. So again, this is not a specific NAFLD gene, but when we consider the genetic impact on the onset, progression, and management of complex disease like NAFLD, we cannot forget to mention this gene and its pharmacogenomic role. But these were just examples. In reality, the interaction between genetic factors, and environmental factors is way more complex. And we have genes like the PNPLA3 that have a specific and straightforward role in promoting lipogenesis and chronic progression. We have genes that have a more articulated role in interacting with environmental factors promoting oxidation. So they promote the oxidation at the mitochondrial level. And we have, of course, genes and epigenetic factors, as the microRNA examples listed here, that interact with the immune system. So interact with the Kupffer cells in the liver, but also with leukocytes at the systemic level. But all these, as you can see, is really intertwined and it's interconnected because one thing induces the other and vice versa. So the complexity level of the pathogenesis of NAFLD really forces us to consider all the genetic networks, all the pathways that are involved, and how individual contributions play in the whole equation with all the others. And just to give you an idea, we mentioned, of course, so far genetic factors, but we forget about epigenetic factors. The microRNAs are just some of those, but they have been clearly associated with the progression from step to step of liver disease, from the normal level to the first stage liver injury, hepatitis, and then fibrosis and cancer. And then just to finish, we must not forget that the genes are also showing us the way for developing new treatments. So this is the translational dimension we were mentioning before, and here is just an example of how drugs may target different nodes of the pathogenic pathway leading to liver disease. At the level of the oxidative stress for antioxidants, at the level of growth factors and other ones on acting on the immune system. So in conclusion, we're living now in an era where we have an increased availability of omic technologies. And the data that are being collected and are currently collected are really suggesting that the implementation of genomic profiling of subjects affected with NAFLD, or even better, at high risk. So we can introduce a level of management at the preclinical stage, are really critical, and they can critically improve the outcome of this disorder. Therefore, we want to apply these to the precision medicine protocols. And the benefit of that would be developing screening protocols for subjects at risk. For example, identifying individuals carrying the PI148M variant in PNPLA3, characterizing the potential biomarkers for the preclinical selection of ideal candidates for specific treatments, and even determining the optimal dosage of these treatments. For example, looking at the variants in CYP2D6 and their pharmacogenomic effect. And another potential benefit is identifying molecular targets for the development of new treatments. For example, inhibiting the HSD17B13 activity through RNA interference. So as you can see, the genetic factors are playing a very important role in the pathogenesis of NAFLD, but they're also providing us with very powerful tools for the management of this disorder. And with this, I'd like to thank you very much for your attention.
Thank you, Professor Boccuto, for your excellent presentation and for perfect timing. For the question, we continue later with free discussion.
Thank you.
Thank you. Now it's my time. So, I am Ludovico Benavoli, you know me. I work in the University Magna Grecia of Catanzaro. Well, I speak to you about non-alcoholic fatty liver disease and Mediterranean diet. When we talk about Mediterranean diet and non-alcoholic fatty liver disease, we are here with a recent past. The definition of NAFLD began 40 years ago. And now we have with 32,000 items in PubMed, and the future is to talk about this new definition of metabolic associated fatty liver disease. In the past, this is the representation of ancient men by Phidia school 5 centuries before Christ. Typical of these statues are typical of my region. The name is Bronze di Riace. And this is the modern representation by Botero. And liver follow the same way, and now the liver is completely full of fat. When we talk about non-alcoholic fatty liver disease, it's important to understand this is an umbrella term that refers to a broad spectrum of lesions, from simple steatosis to steatohepatitis, fibrosis, cirrhosis, and finally the complication like to hepatocellular carcinoma. The pathogenesis of this condition is a multi-step process, and the central role is played by insulin resistance with oxidative stress and free radical production, necrosis, fibrosis, and finally steatohepatitis. However, in the last 2 decades is also arrived new concept like to intestinal permeability, leaky gut, and inflammation by portal blood flow. The epidemiology of NAFLD is increased in the last time. For example, in Europe, it's estimated that 25% of general population present fatty liver. And of course, this is the classic evolution, the classic natural history of NAFLD, similar to other liver disease. However, recent retrospective data report that it's possible the evolution from simple steatosis or steatohepatitis to the final step like to hepatocellular carcinoma. This is important to understand that in the near future, the main part of liver transplantation will be made in patients with metabolic cirrhosis. Another more important concept is the concept linked to gut microbiota. This 2 kilograms of weight of our body, 3 million of microbial genes, 150 times more genes than in the human genome. For this reason, we talk about Homo bactericus. Everyone has a peculiar gut microbiota. Everyone harbors unique strain. And the microbiota is an organ, a dynamic organ that follow the evolution of our personal history, influenced by type of nutrition, healthy, unhealthy status, presence of disease, malnutrition, antibiotic treatment, and metabolic condition. And also, of course, the evolution during our different period of life. However, when arrive a disequilibrium of our microbiota, this condition is called the gut dysbiosis. And now we know that gut dysbiosis is linked to several diseases, not only autoimmune disease, but also chronic condition like to liver disease. In fact, in patients with NAFLD, is usually to found, like to reported in this interesting recent review published by Hepatoma Research, different bacteria, in particular Firmicutes and gram-negative bacteria that produce increasing in gut permeability, leaky gut condition, and the presence of lipopolysaccharide in portal blood flow with consequent inflammation of the liver. In this context, an healthy diet like to Mediterranean diet that presents polyphenols, high quality fats, micronutrients, and fibers will determine the beneficial property maintaining gut eusbiosis and intestinal integrity. However, Western diet characterized by high levels of salt, unhealthy additive, and excessive red and processed meat consumption have a poor effect on intestinal barrier, on gut microbiota life, and and induce local inflammation and the presence of LPS in the portal bloodstream that contribute to chronic inflammation of the liver. In this condition, diet, lifestyle, environmental factor, and genetic predisposition play a pivotal role to detect NAFLD population, in which a genetic or not a genetic predisposition predispose the subject to development of NAFLD and NASH. In this context, it is important to report the role of gut dysbiosis with portal circulation present of cytotoxin and LPS, and the role played by inflammatory cytokine produced by central fat mass. Of course, now we live in the omics era. Until now, our concept of the medicine is that genotype induces disease and finally phenotype. But the last recent data report that diet composition influence our nutritional status, and consequently nutritional status influence our phenotype. But not only, also nutritional status is can influence the genotype predisposition and the genotype development of the disease, and the genotype also influence nutrition phenotype and also the type of our diet. Well, the natural history of NAFLD development is clear, and the diet play an important role to cut this natural history. It's important to report to you a new concept, the concept of functional foods. These are the foods defined as whole food along with fortified, enriched, or enhanced food that have potentially benefit effect on health when consumed as a part of varied diet or regular basis and effective level. This is the definition of Academy of Nutrition and Dietetics. In this context, I report to you some examples of functional food. The first one is olive oil. For example, Italy is the second world producer of olive oil. And olive oil is well known to present a high presence of polyphenols, antioxidant, and omega-3 saturated fats. And these molecules play an important role on liver disease in general, in fatty liver disease in particular. In fact, polyphenols and omega-3 present in the olive oil induce immunomodulatory effect on hepatocyte cells, decrease of oxidative stress present in the fatty liver, an anti-inflammatory and antioxidant effect in the systemic in our system with a reduction of inflammatory cytokines, modulation of DNA presentation, and reduction of peroxidation of lipids inside of hepatocyte. The second one is bergamot. Bergamot is a fruit-like to citrus, typical of Italian region, produced in this part of Italy, is the point of our country, in a space limited of 200 kilometers. Bergamot is used for cosmetic industries. However, many studies, in particular this study made by Parafat et al., reported that the use of bergamot included in a healthy diet reduce the fat accumulation in the preclinical study and improve the levels of transamino acids of gamma GT, improve also insulin resistance and the fat level in the blood of mouse in a preclinical context. Another one are ancient wheats. Ancient wheats arrive in the south of Europe, in Italy, many centuries ago, when the men arrived for immigration to the fertile crescent in Asia. However, Ansel wheat, in particular, Khorasan wheat, improve biochemical profile, oxidative stress parameters, inflammatory parameters when it is used in the diet for a minimum of 3 months. Well, the influence of diet in the pathogenesis of NAFLD has been reported, and the cornerstone in the management of NAFLD implies a dietary modification to decrease body weight and to increase the physical exercise. And to begin this process, it's important to adopt a diet regime rich in monounsaturated fatty acid and omega-3, fruit, fibers, vegetables, with a reduced intake of saturated fat. And these are characteristics typical of the Mediterranean diet. In fact, a multidisciplinary approach is necessary in patients with metabolic disease in general, and in particular with non-alcoholic fatty liver disease, with personalized diet, personalized physical activity supervisor, if necessary, motivational support, and long-term monitoring program. The dietary recommendation is to reduce the introduction of calorie in daily diet. Ideal diet is represented by 50% of carbohydrates, 30% of lipids, and 20% of protein. Diets are designed to create a calorie deficit around 1,000 kilocalories per day and producing a weight loss of maximum 1 kilo per week. And these characteristics are typical of Mediterranean diet. Mediterranean diet was defined for the first time from Ansel Keys in 1957 in Nicotera. Nicotera is a small village like to Vibo Valencia in the the Poro mountain in the south of Italy. And these are the characteristics of Mediterranean diet with this pyramid that in 2010 are adopted by United Nations like World Heritage of Humanity. It's important to underline the area of Mediterranean Sea. Mediterranean area is 1.6 of the world length. However, in the Mediterranean area are present 30,000 plant species like to Amazonia area, 10% of plants worldwide with flowers, same plant diversity of tropical Africa, 0.7% of sea water, and around 8-9% of no marine organism. And this is our typical dish, the typical dish of Mediterranean diet, with fruit, protein, in particular vegetable proteins, and grain, rice, and pasta. 1 glass of red wine for dish if no disease are present, and 1-2 servings of olive oil. The effect of Mediterranean diet on NAFLD is direct. In fact, the Mediterranean diet induces reduction of overweight, reduction of hepatic fat accumulation, improvement of inflammatory status, equilibrium of our gut microbiota, reduction of insulin sensitivity. And in this slide, you can see the typical Mediterranean diet like to be defined in the first pilot study made in Nicotera in 1916, rich in cereal, legumes, potatoes, and with just a small intake of animal protein. In fact, the people that follow Mediterranean diet present a reduced risk to develop cardiovascular diseases like to be defined in the 7-country study, the study of the more important published by Ansel Keys. And it's important to report to you the MAI. MAI is Mediterranean Adequacy Index, an index that reports the adherence to this healthy diet. In fact, many international guidelines to EASL, EASD, and EASO clinical practice guidelines, and American Association of Clinical Endocrinology guidelines report that Mediterranean diet is the first and more important and more effective diet to be used to treat patients with non-alcoholic fatty liver disease. Not only this, but also Mediterranean diet is helpful to prevent the development of cancer, like to hepatocellular carcinoma and colorectal cancer. In this way, I report to you the message made during the Expo of Milano, during the Mediterranean Diet Week, that is to improve the Mediterranean diet, the natural way for health. Finally, the take-home message of my speech are the influence of the diet in the pathogenesis of NAFLD has been demonstrated. Considered the worldwide preference of NAFLD, the definition of therapeutic algorithm is the challenge in this field of the near future, and in this way, the new definition of NAFLD can be useful. The cornerstone in the clinical management of NAFLD include two easy and pivotal concepts that are dietary modification associated with improvement of physical activity. In the era of evidence-based medicine, Mediterranean diet can be considered the gold standard in preventive medicine targeting NAFLD. I want to be grateful to my colleague of many other universities worldwide that contribute to make this presentation. Thank you from me and my study group to Catanzaro. Good, thank you. And the free discussion is open about the lecture of Professor Luigi Boccuto and my lecture. If you have a question, please report in the chat to be reported to the speaker.
I have a question. You mentioned Nicotera. It is the beautiful village on the coast. So the diet there was quite, calorie poor. And Ansel Keys, you showed, he didn't, originally he started with Nicotera, but then he left it out. In his 7-country study, Nicotera doesn't appear anymore. Can you say, you know that why? Why was that?
Yes, thank you for the question. Nicotera is a very small village in our region, and after the Second World War, represented the typical village of Italian tradition. For this reason, Ansel Keys arrived in Italy and during his presence, detect that in this area the age of the subject is very high, reported to other countries, and that in this population don't found a limited number of patients with chronic cardiovascular diseases. However, in this first step, he studied this population. However, for the problems with found, the study in Nicotera are not continued. And for this reason, in the 7-country studies that you report is a very good and important study, are being included other villages and other cities of Italian context, like to Cilento or like to Rome. However, in this moment, this type of diet now in Calabria, where the region where this diet was detected, defined for the first time, is very limited. And like to other European and Western world, we know a diet founded on junk food. And this is a problem also because our population, in particular young people, are obese. For example, Calabria and Campania, our south region, are the region with the high percentage of obese child in Europe. And this is a big problem. Thank you for your question.
Thank you.
Professor Rouabia from Algeria reported, asked, in many times patients with NAFLD have comorbidity treated by drugs, like diabetes patients. What about treating two conditions with one drug, like in diabetes, metformin or liraglutide? I replied to Professor Rouabia. Yes, one treatment is ideal. However, until now, international guidelines report that the only strategy, the only approach to treat this type of condition is represented by lifestyle change. We hope in the near future, with many phase 2 and phase 3 studies that are coming, to have new and many treatment to approach this type of patients, like to obeticholic acid and other that we can arrive in the near future. However, until now, FDA and EMA don't approve no treatment. Thank you for your question. And I want to ask to Professor Boccuto for his excellent presentation. What do you think about the application of this recent study in clinical practice? From bench to bedside, what is the near future for our clinical practice? Thank you.
Thank you for the question. It's a very important one. And I think that it has different impact on different healthcare systems. For example, a healthcare system like here in the United States, where the private insurance component is very important, has to quantify the financial impact of genomic stratification of the population. For example, screening individuals carrying the PNPLA3 frequent variant, the I148M, might prevent hospitalization and lifelong treatment for a lot of patients, because it might help us to work on the environmental factors. So introducing a healthier diet, a healthier life hygiene, drugs that will prevent the accumulation and the promotion of lipogenesis might have a dramatic positive impact on the health of the individuals, but also on the healthcare cost. In general, it's a matter of spreading the culture of preventing liver disease by acting on the modifiable effects. We're on the edge of genome editing with CRISPR-Cas and other editing technologies. And so soon enough, we should be able to change also the genetic factors. But for now, I believe that implementing genomic profiling, implementing the culture of prevention, like working on the diet, as you mentioned in your presentation, Professor Benaboli, is going to be critical, because it will introduce a new approach, not only to the liver disease, but to the metabolic disease in general.
Thank you, Professor Boccuto. If you don't have other question, no. Okay, I want to invite Professor Giuliano Ramadori, Executive Chief Editor of Hepatoma Research, to introduce to present the journal Hepatoma Research. Please, Professor Ramadori.
I apologize for the fact that I don't have slides, but I want just to mention that I am very much pleased to be, let's say, the chief editor of the journal, and I'm very much pleased that we have introduced these kind of seminars like these wonderful seminars today, and we are pushing knowledge, spreading knowledge around the world. Thank you for your contribution, and we also like to be as much as possible original and critical in all the contributions. So I am very happy that you mentioned, for example, the Mediterranean diet, and you mentioned that it started in Calabria. Nicotera is Calabria, but Ansel Keys was he came from from the United States, and established his second home in Acciaroli, in Cilento. Now Victoria just provided me with the slides. Can you give me the next one, Victoria? And this is the editor-in-chief, Professor Cao, and myself. Next slide. The aim of the journal, I mentioned before, we want to cover the all field of cell biology and all the clinical field of hepatocellular carcinoma and cholangiocarcinoma of the liver. And of course, genetics and pharmacology, immunology are also very important topics. The next slide. You can see here the readership of the journal, which is now 7 years old. And many readers are in the United States, but also in the UK. Next slide. Next, Victoria. The journal doesn't have yet any impact factor, but you can see that the views and downloads, thanks to the contributions of many scientists and components of the editorial board, is increasing every year, and significantly. The next slide. These are members of the editorial board, for example, from China, Singapore, and the United States. Next slide. It's of course very important to have Chinese researchers in the editorial board. China is a large country with many patients with hepatocellular carcinoma. That's why it's important, and we are aiming to get more and more scientists involved in the journal. And here are the countries of the editorial board members, and you can see here, Italy is very well represented, and we thank all the members from Italy for their contributions to the journal. Next slide. The journal is, of course, very interested in having special issues and having members of the editorial board leading, the projects of the special issues. You see here Guido Gerken is one of the most active in Germany. Here, Professor Gerken from Essen. Next slide. These are ongoing special issues, and I would like to you to contribute an article in one of these special issues. Here you see here now the special issue of our chairman of today, Diet, Liver Disease, Liver Cancer Prevention from the Source. He mentioned, olive oil as a protective factor in the diet. He didn't mention a lot the good wines from Calabria. I like a lot, but of course, not much is allowed in the diet. Next slide. And these are the web seminars. I found the web seminars of today very successful. As we had several in the past, and we will have also in the future. So we will have the next one is I believe it's on the 17th of this month, and I would like you to be a participant of this next one. And next will be in January. And these of course are the web seminars are free for participation. And we had, as I mentioned already in the past, very nice and important web seminars. The journal also, Victoria showed me on the next slide, also admit video abstracts of the manuscript you can contribute, which is an additional way to stress the contents of the articles. Next one. Very important topic is still, as we heard today, the topic of the gut microbiota and of the bacteria being part of the pathology in the development of liver disease. And what's next? Next, Victoria. So, of course, the journal is very much interested in making your articles interesting for readers. For example, they are using, I'm seeing Victoria is using quite often LinkedIn. You will have your contribution presented in another way in this platform. The Patoma Research is seeking support and of partners, and is partner of ILCA and other organizations in the world. And Victoria, you know more than I know here on this topic. The advantage of the Patoma Research is fast and rigorous peer review to improve the quality of our publication, of your publication. You can have also different forms of discounts when you want to publish a manuscript. And your paper will be promoted in different ways. And the journal offers web seminars, and you can you are welcome to organize in the future such web seminars as Dr. Abenavoli did today. So I think this should be the last one slide. This is encourage the journal, encourage of course to send manuscript and to publish with us, with this Hepatoma Research. And good. I was not very much very good prepared, Victoria, but thank you for helping me. And so that's it's all what I can and want to say about the journal. And of course, I thank Professor Abenaboli for the efforts that he put in the organization. I know it's not that easy as it can appear to get well-known scientists to deliver a speech and to stay this afternoon with us. And I enjoyed, I have to say, the mentioning of the Mediterranean diet. I can tell you, Professor Abenaboli, that I was born in Monte Giorgio. And if you look at the 7-country study, Monte Giorgio was one of the village, you know, where 760 men were chosen for participation in the study. And one of the men of the study was my grandfather. So, you know, I enjoyed very much listening to your talk.
Thank you, Professor
Thank you.
Ravada. Thank you. Thank you for your the presentation of the journal. It's very important to be supported to make the science, and Hepatoma Research achieve a big aim that is to connect many scientists, scholars, and students, and PhD, and more people around the world. It's more important. Thank you to you for your executive work, and to Hepatoma Research for this important platform and for this opportunity. Well, we are perfectly in time, and I invite my friend Professor Emilio Scarpellini, researcher to University of Leuven in Belgium, to present his speech, Gut Microbiota and Hepatocellular Carcinoma Development. Please, Emilio.
Hi to everybody. Just a second. Yeah, here I am. So first of all, thank to Ludovico for inviting me in, to all the editorial board for this exciting opportunity. Today we'll try to speak about the gut microbiota and hepatocellular carcinoma natural history and development in particular. What is gut microbiota? We can say that it's the largest, the biggest symbiotic ecosystem within our body. It's about 2 kilos weight, and is not only composed by bacteria, but also from other microorganisms like archaea, protozoa, yeasts, and viruses, most of all. It's about several trillion of microbes. And the main bacterial components of our microbiota are Firmicutes, Bacteroidetes, Proteobacteria, Actinobacteria, Verrucomicrobia, Fusobacteria, and Cyanobacteria. Our human health is affected from a multidimensional point of view by these bacterial phyla. And as you can see here, these are the main one divided by gram-negative and gram-positive. What are the functions? We describe it briefly in here, as my colleagues did partly before. The functions of gut microbiota in our body. As you can see, they contribute to the absorption of micro and macro nutrients like proteins, fibers, fat digestion, and they participate in the biosynthesis of neurotransmitters, BCAA, short-chain fatty acids, triglycerides and fatty acids, and vitamins as well, neurotransmitters, transmitters, and hormones as well. So they affect the functioning of our brain, of our circulatory systems, the adipose tissue in a bidirectional way, kidneys, and so on. So this is a little bit of taxonomy, just to remind that about the bacterial components of our gut microbiota. We can recognize phylum, family, genus, and species. And here, among the main phylum, we remind and we record Firmicutes, Bacteroidetes, and Actinobacteria. And this is a nice picture where we need legend just to understand what's going on. So from neonate infant until our old people, we can see that the amount of represented phyla of bacteria, they change dynamically. So the beta diversity, that it's the difference of microbial composition between subjects, the inter-individual variability, is going down when we become older and older. Meanwhile, the bacterial diversity should increase, so the alpha diversity, but towards several ways of alteration of the composition of our microbiota. And why it happens? As my colleagues already mentioned, there are age-specific factors, genetic factors, and Professor Boccuto in detail depicted this very complex relationship, let's say, between microbiota and the host and environment and so on. And we see that here we have different factors that can be comorbidities like obesity, type 2 diabetes, that affect the composition of our microbiota, not excluding the aging. So far, how does it work, our gut microbiota? And it helps us to understand how it interacts with the liver. So in our gut, there is fermentation of carbohydrates, fibers. So they access through the blood stream to the liver. They become metabolized. But this happens not only for nutrients of any origin, but also for toxic substances. So also for our bile acids. So the 1-2% of enterocholic recirculation of our bile acids is a small amount in consideration of all the amount of bile acids that we produce circulates not only in the gut. But this small amount condition gut microbiota composition and all and most of the metabolic processes happening within the liver and in the rest of the body. And of course, our gut microbiota contributes with antigens presentation, the gut, and the systemic immune system education to the development, maintainment of immunity, tolerance, and in disease presentation. So what happens when our gut, liver, let's say immune axis works? Well, what we say. So here there is our normal gut microbiota or eubiosis condition. Tight junction regulate intestinal permeability to antigens, to toxic substances, but mainly to antigens. So a few of them will be presented to the immune system through the portal vein circulation into the liver, Kupffer cells, stellate cells, TLR4. When there is gut microbiota dysbiosis, there is an increased or impaired, let's say, intestinal permeability. Pumps, dumps, they pass through blood circulation, portal circulation into the liver. There is an hyper and not physiological activation of our immune system. There is inflammation, fat deposition, inflammation all together. They may lead to cirrhosis and to HCC development. We can jump from NASH to HCC, like my colleagues previously reminded. And in detail, what happens? That these dysbiosis affect bile acids metabolism, ethanol production with hyperproduction and altered metabolism, choline providing to our hosts intestinal permeability, so perpetuating this vicious circle responsible for our liver disease. And this is an example, a very nice figure from this very nice review, very updated by XU, that shows how every liver disease has a proper dysbiosis. So alcoholic fatty liver disease, NAFLD, NASH, and MAFLD, the bigger entity that we are coming to get in touch and playing with, the hepatocellular carcinoma, and also the autoimmune hepatic disease, including the cholangiopathys. And this is what we did with Ludovico very recently. We had a chance to publish this work, trying to study ex vivo what happens to gut microbiota dysbiosis in a cohort of cirrhotic and non-cirrhotic patients from our center here in Marche. And what we saw, analyzing with the anthropometric and patient characteristic, that the two groups of cirrhotic and non-cirrhotic, they were quite similar. There was a very mild difference about autoimmune or cholangiopathys representation, but was not statistically significant according to the sample size. What it was interesting and quite overlapping and so confirming literature data, there was a significant prevalence of small intestinal bacterial overgrowth in both cirrhotics and non-cirrhotic patients versus healthy volunteers. And this data was more significant for hepatic encephalopathic cirrhotic patients. What we saw dosing endotoxin levels is that there was as well a significant difference of cirrhotic and non-cirrhotic patients versus healthy volunteers. But this difference was even more significant according to the gravity, so the stage of gravity of liver disease, according to Child-Pugh status. Looking, and this is the most important slide, at the eventual correlation or relationship between small intestinal bacterial overgrowth and blood concentration of endotoxin, we saw the very interesting result. That is this, that SIBO absence is not compulsory for absence of higher levels of endotoxin. So probably the small intestinal bacterial overgrowth describe a very upper GI tract gut dysbiosis. That does not describe the passage through an altered intestinal permeability of endotoxin, so lipopolysaccharide, that is a pump. So these are the study linking in animal models of liver disease, so cirrhotic and non-cirrhotic, with or without hepatocellular carcinoma, gut dysbiosis, with occurrence of liver disease, occurrence of HCC. This is the rest of the slide, and here we can clearly see that there is a correlation between dysbiosis. The dysbiosis can be different according to the different model of liver disease use, either inflammatory, either diet linked with the high-fat diet, or toxic model of liver disease. What about human data? Are they in? Yes, there are several. So this is a solid evidence as well. And we see that even here, with or without HCC development, with or without liver cirrhosis, there is a consolidated literature data showing that there is an heterogeneous gut dysbiosis according to the presence of liver disease, to its stage, and or to the presence or absence of hepatocellular carcinoma. What about dysbiosis and liver regeneration? Is there a relationship? Yes, it's there. So we see that the regeneration of the liver is affected by several cytokines, hormonal, not hormonal agents. And then there is a termination phase, mainly affected from TGF-beta. What happens in an animal model where we operate a partial hepatectomy? That there is a variation, a very early variation in terms of days or hours of our microbiota. So we can imagine what happens, the shift that happens, especially in the liver cirrhosis patients that are developing or not developing hepatocellular carcinoma. And this is one of the most nice study showing in an inflammatory model of liver disease leading to hepatocellular carcinoma. What happens? So in terms of dysbiosis, we see that there is during liver inflammation, so we can imagine an ASH patient, for example, a microbial gut invasion of the intestinal mucosa, so they pass the altered intestinal permeability, and we arrive until liver cirrhosis. Here there is an higher lipopolysaccharide biosynthesis. And it's interesting that our HCC get nourishment from not only from carbohydrates, but starts the switch from proteins. So also the metabolome within our liver and our intestine changes. What about systemic inflammatory response? Here you can see lipopolysaccharide levels going up, and you see our systemic immune response that beforehand was very intense on the GI tract and also in the liver going down, and it increases our inflammatory response within the liver. So the tolerance is going down, so the surveillance towards possible hepatic cells that under this push can get mutation and become hepatocellular carcinoma. So that's what happens inside the liver, like we were saying. Oh, the inflammatory response works. No, and now it doesn't work anymore. So our T8 cytotoxic lymphocyte, they go down. Then they don't get surveillance towards our hepatocellular potential cells over proliferation. But this is an active process. In this very, very nice model of inflammatory liver disease, we can see what? That deleting the NLRP6 NFKB control gene in these mice. So this is the group. We have a significant and dramatic shift at 13 weeks time point of gut microbiota. And in particular, Akkermansia mucinifila goes down in terms of abundance. Intestinal permeability in this kind of modified mice towards the control model increase. And what happens if the intestinal permeability increase? You see these deleted mice, the tumor nodule size significantly increase. Further, you see that according to the presence or absence of NLRP6 deletion, it increases the amount of the mesenchymal monocytic stem cell suppressor that go to suppress our cytotoxic T cell CD8, and you can see clearly. And you see the inverse correlation. The more our mesenchymal cell increase within the liver, the lower it becomes the concentration of the tolerance cell, let's say, that maintain surveillance towards the tumor cells. What happens if we give antibiotics cocktail to these mice? We see that significantly these suppressor cells go down, and we see that in case of these NEMO modified mice, this is not true anymore. What happens if we operate a fecal microbiota transplantation of NLRP6 deleted mice to the, let's say, control group, the NEMO delta EPA group of mice? That it is reverted the concentration of suppressor cell, and the cytotoxic T8 cell, they go down as well here. But very nicely, that what it happens as well in terms of abundance of single phyla of bacteria. So if, as we operate the fecal microbiota transplantation, the quote of suppressing cells or CD8 cytotoxic cells increase, and the abundance of Akkermansia mucinifila goes down. What in terms of treatment response? So, are there bad bugs for response to anti-androgenetic and checkpoint inhibitors in HCC evidences? Yes, there are several. So these dysbiosis affect the response to several chemotherapy in treatment of HCC. At this point, most of us know that in comparison with cholangiocarcinoma, in cholangiocarcinoma, we know about 40% of mutation. So we can more easily use checkpoint inhibitors, drugs, just to prolong survival of these patients, to improve their quality of life. And we hope to reduce volume and to improve expectancy of life for this patient, at least in the next 5 to 10 years. In terms of knowledge of HCC, we don't know almost anything about the mutation. We know about, in comparison with cholangiocarcinoma, 2.5% of mutation. So we will need a biomarker that allow us to make an early cancer diagnosis and to predict treatment response, especially with the newer and newer checkpoint inhibitors that have been used in the last years for HCC treatment. Are there evidences? This is a very nice Chinese study where we see that we start to see the mutation of different Escherichia coli, in particular strain with this marker here below, and we can get a prediction of early hepatocellular carcinoma presence, so diagnosis, and response to chemotherapy, because we know that alpha-fetoprotein and other biomarkers derived from alpha-fetoprotein very often, let's say in 40–50% of cases, they don't allow to get even a suspect diagnosis of early HCC. What about gut microbiota and other biomarkers and response to treatment? Do we have other data? Yeah, that's a very nice study from my ex-university, so Catholic University of Sacred Heart, Gemelli Hospital, and that's the friend Francesca Ponziani. They performed a small study, but very nice, where mechanistically they saw gut dysbiosis improving and fecal calprotectin concentration decreasing in those patients getting a good response and tumor diameter reduction after immunocheckpoint inhibitors usage. And what is the mechanism? Well, the mechanism through which modulation of gut microbiota with antibiotics, probiotics, let's say also postbiotics, we are in the age, and metabolomics and so on, and the omics, as Professor Bocuto magistrally said, are different, so we can act on bile acid metabolism, on immune response, short-chain fatty acids synthesis, cytokines synthesis, and factors that regulate hepatocytes mitosis and apoptosis. Is a gut microbiota target for HCC modulation? Are there evidences? Well, yes. There are several evidences uses both probiotics at a very high concentration, antibiotics cocktails that are showing some data, very interesting data, at least on a reversal of dysbiosis and improvement of HCC behavior. What about men or human studies? Where there are still a few, and there is some first hint on the significant association between gut dysbiosis shift towards eubiosis and faster or better reduction of hepatocellular carcinoma dimensions, improvement of patient and liver function. So what are our concluding remarks to make a summary? That we have a different gut microbiota composition, not necessarily we can speak about dysbiosis in every hepatic disease at any stage. The dysbiosis is of course involved in pathophysiology of hepatocellular carcinoma. We focus on the gut-liver immune axis. The dysbiosis has been confirmed to be present in different animal models and human studies about HCC development. Gut dysbiosis, and this data is crucial for me, is responsible for the immune response switch within the liver, both for tumor initiation and progression. Certain gut dysbiosis subset hallmark are associated with certain HCC responses to both anti-androgenetic and checkpoint inhibitors agents. Gut microbiota can be used as a marker, perhaps, for early HCC diagnosis and treatment response prediction. Yes, we have the first promising data. Finally, the gut microbiota modulation show promising first results in humans and also according to animal data as being an add-on treatment together with the chemotherapy. And the last one, the most important slide for me, I thank you for your attention.
Thank you, Emilio, for your excellent presentation. Maybe at the end of this session, we can speak about other interesting points and to reply to different aspects.
Yeah.
I continue to invite Professor Yasser Fouad, Professor of Gastroenterology at Minya University of Egypt, President and founder of the Egyptian MAFLD Research Group, to present his speech entitled Diet and MAFLD. Please, Professor Yasser.
I hope you hear me now. Are you hearing me?
Yes.
Yes, please.
Are you hearing me now?
Please, Emilio,
Yeah.
stop your presentation.
Yeah.
Yeah, thank you.
Okay.
Please,
Ahmed,
yes,
yes, I'm here. Now I should share my screen. All right. Is it okay now? Are you hearing me?
Yes, yes, yes.
Okay.
Everything is
thank you.
okay.
Oh, thank you very much. Thank you, Hepatoma Research, and thank you too. Thank you, organizing committee. I really enjoyed the lectures this night, and I hope everyone is awake now. I will go fast because of the time. Diet and MAFLD, I have nothing to disclose, but as you know all that prevalence of MAFLD or NAFLD is increasingly all over the 30 years to reach about 30% of population worldwide. And we have in the Middle East region the highest prevalence all over the world. It's approaching about 40% of the population. So recently, MAFLD, metabolic associated fatty liver disease, was introduced by Islam et al. 2 years ago, and it's gaining importance because it's a lot of information, research, and debates all over the last years. So what is MAFLD? MAFLD is a metabolic associated fatty liver disease. It's a hepatic steatosis in any patient. It's detected by ultrasound or by liver biopsy in presence of obesity or overweight patient, or in presence of diabetes mellitus, or 2 out of 7 metabolic risk abnormalities. Here we diagnose MAFLD by inclusion criteria. And this is the difference between MAFLD and the older name NAFLD. NAFLD was dependent on exclusion of all other secondary causes of liver disease. So now we are including the parameters of MAFLD syndrome or diabetes and make this diagnosis of fatty liver disease easier. And as we noticed in this paper recently published, all MAFLD patients nearly having NAFLD by old definition, except for few patients with no other factors of liver disease who are diagnosed as non-MAFLD NAFLD. It means that all patients nearly with MAFLD are the older patient with NAFLD. So tremendous amount of research all over the last 2 years proved the superiority of MAFLD criteria versus NAFLD criteria, as you see here in this slide, in detecting a higher incidence of HCC also predicting the extrahepatic malignancy. And MUFFLED criteria was found better than NAFLD in identifying patients at high risk of renal disease. MUFFLED also criteria changes identified a large proportion of subjects in this huge number of patients in Korean subjects who had a higher risk of developing cardiovascular disease. MUFFLED identified severity of coexistence of fatty liver disease with other liver diseases in these two nice papers. Continuously also evidence proved that MUFFLED criteria outperform NAFLD criteria in identifying patients at high risk of hepatic fibrosis. And MUFFLED demonstrated high diagnostic ability of fatty liver index in the detection of hepatic steatosis. It was found that FIB4 and NAFLD fibrosis score could confidentially be used to exclude advanced fibrosis in overweight, obese, and severely obese patients with MUFFLED. Renaming it to MUFFLED increased the awareness of disease among primary care providers and physicians in other specialties. And positive impact on clinical trials was seen nicely in published two papers. So many societies worldwide endorsed the MUFFLED criteria, including APASL, Middle East, MENA region, Latin American Association, Study of Liver Disease, and Sub-Saharan African Position Statement, continuous Chinese Society of Hepatology Position Statement, and groups of nurses, groups of patients, and International Society of Tropical, and many other societies now are endorsing. And a paper published recently in Lancet Gastroenterology with signature of more than 1,000 hepatologists all over the world from more than 130 countries. So it's a well-established name, well-established literature, well-established research, and it's now going a consensus by AASLD and EASL finally to endorse this name or discuss this name. So going back to MUFFLED, since no approved medication till now by FDA to treat MUFFLED patients, we only have lifestyle modification as the treatment of choice available in our hand in management of MUFFLED patients. And lifestyle modification include weight loss mainly through diet control, exercise, and we can add that coffee intake as a lifestyle is one of the interesting changes of life to manage or add it to management of NAFLD. All of us know this pyramid very well, and this pyramid shows nicely that if you lose more than 10% of your weight and you have a fibrosis, this will reverse the fibrosis in the liver. And even if you lose only 3% of your weight, steatosis will be reversed in your liver. So losing weight proved by evidence that it's important to reduce the steatosis and steatohepatitis and fibrosis. But the problem is that people who lose weight, only minority of them will sustain losing weight after the first year. By evidence, it's only 10% of patients who will lose weight beyond the first year and sustain this. So there is a multidisciplinary approach to maintain losing weight, including psychiatrists, family health, and hepatologists. So what diet is important? What diet is convenient to patients with fatty liver disease? If you look at these societies nearly covering the whole world, EASL, ESP, EN, AASLD, APASL, AGA, and so on. They are all considering that the hypocaloric diet is the main or the important diet to manage NAFLD patient. It depends on reduction of diet by 500 to 1,000 of calories per day, targeting to lose about 0.5 to 1 kilograms per week. Most societies also considering that fructose avoidance is important in management of NAFLD, and they encourage the coffee intake, and they encourage reduction of the amount or abstinence of alcohol in some situations and some studies. So MUFFLED treatment with diet is important through the mechanisms of weight loss, improving insulin sensitivity, reduction of hepatic steatosis, and reduction of steatohepatitis. So which diet? Is it low carb or low fat? There are many studies focusing on comparison between low fat and low carb in management of NAFLD. And actually, there is a minor difference in favor of low carb diet to manage NAFLD. There is initial promise in consistent results between both, and losing weight here is the key. It's unclear whether the type of diet is important, but actually low carb seems better than low fat in management NAFLD. Ancient times in Egypt, if you go to all the Egyptians, they used to have foie gras. That means that they overfeed the duck or geese to get a fat liver or fatty liver, which was delicious to eat. If we look at the human, they are doing themselves this. If you eat so much carbohydrate, you will get fatty liver, and this depends on that. So carbohydrates are important, and this is not what you eat only, it's also the matter how you eat. So eating snacks is dangerous. And more snacks associated with sugar are also associated with increasing in the intrahepatic triglyceride content. So snacks between main meals are associated with steatosis, as proved in this nice paper. Sugar may increase the insulin level, and insulin level is associated in the study with the steatohepatitis or NASH, and this was published recently in Gastroenterology. And this nice study from JAMA network comparing the low sugar diet versus usual diet in adolescent, it found that the mean increase in hepatic steatosis from baseline to week 8 was significantly greater for the intervention diet group with low sugar diet, from 25% to 17% compared to the usual diet group. So in meta-analysis, they found that the meta-analysis of 10 international clinical trials of low-carb diet is than 50% in patients with NAFLD. 10 studies evaluated ALT and 9 evaluated AST, 5 studies evaluated GGT, and 4 evaluated intrahepatic lipid content. The result was that carbohydrates diet significant reduction in intrahepatic contents, but no significant reduction in ALT, AST. What about non-nutritive sweeteners? There's a lot of debate about this, but they are epidemiologically associated with obesity and type 2 diabetes. And this explained by that the patient with diabetes or obesity are encouraged to take some non-nutritive sweeteners. But this in ALT from Framingham Heart Study cohort, diet soda not associated with elevated ALT after adjusting for BMI. Sugar sweetened beverages significantly associated with elevated ALT in a dose-dependent manner, including after adjusting for BMI. But non-nutritive sweetener may induce metabolic dysfunction through activation of sweet-taste receptors by change in taste preferences, increase the insulin secretion, and increase the appetite in some patients. It may alter also the microbiome and specific effect, particular for aspartame and sucralose. So it's a debate to encourage or not to encourage patients to take non-nutritive asides. What about popular diet? Mediterranean diets mentioned very nicely in the lecture in this slide. And Mediterranean diet has a very good evidence to use and endorsed by some societies now in treatment or in a line of management or in lifestyle modification in patients with NAFLD. And ketogenic diets are also studied in very few studies, but has some limitations, and because it's association with the lipid increase. DASH, the diet approach to stop hypertension and focusing mainly in vegetables and fruits with some proteins. It has a problem with tolerance from the patient. Intermittent fasting, either daily by 8 versus 16 hours, 8-hour eating and 16-hour fasting. Another approach by 2 days per week fasting or day after day fasting. That's also not tolerated by many patients, and a recent published paper in New England Journal of Medicine a few months ago proved that there is no value of intermittent fasting in treatment of patients with obesity. There is no evidence that the most popular diets, except for Mediterranean diet, are important for management of MAFLD patients. So what about micronutrients or vitamins? Vitamin levels, the liver is involved in transport and storage of many micronutrients. In general, patients with NAFLD have lower blood levels of zinc, copper, vitamin A, vitamin D, vitamin E. So low levels of these micronutrients associated with NAFLD severity, although the mechanisms remain unclear. So vitamin D supplement, I think that the only vitamin used in NAFLD patient and endorsed by some societies is vitamin E. Vitamin E can be used in treatment of non-cirrhotic patients with NAFLD and in patients with biopsy-proven NASH at a dose of 800 international units per day, and trying to check for prostate, because prostate cancer is one of the complications of long-term use of vitamin E, in addition of dialysis also. So when you use vitamin D for a long time, you can check in males for the prostatic changes. So to summarize, dietary habits or dietary management in patients with NAFLD, low dietary sugar, there is a theoretical reason to limit sugar, especially fructose or processed sugar. Non-nutritive sweeteners, there are theoretical reasons to avoid, and in low caloric, low fat or low carb diet, no diet has consistent superiority, although there is some differences, provided simple sugars and total calories are less. Popular diets individualized to patient preference and to consider Mediterranean diet according to some societies. Vitamin supplementation, vitamin E is recommended for non-diabetic adults with NAFLD, but consider risk of complication as I have mentioned before. So what about coffee? Coffee intake. There is a nice meta-analysis published recently showed 2 case control studies, 8 cross-sectional studies, and 1 prospective cohort study included in this. Coffee consumption was significantly associated with a 35% decreased odds of significant liver fibrosis, although it was not associated with change in prevalence or incidence of NAFLD, but significant liver fibrosis reduced, and this was not in NAFLD only. Previous studies showed that coffee intake may be beneficial in other causes of liver fibrosis or liver cirrhosis as chronic hepatitis. What about polyunsaturated fatty acids? It has been shown that reduced intrahepatic triglycerides noticed with PUFA consumption. Also, steatohepatitis improved and insulin resistance improved. The metabolic benefit observed for monounsaturated fatty acid also shown with some cases of in some studies. And we have to remember that saturated fatty acids increase the intrahepatic content at reverse. They increase the steatosis and they increase steatohepatitis. So we may consider PUFA, fish oil, and MUFA, olive oil, in the diet management of patients with NAFLD. MAFLD consider the presence of other etiologies. So MAFLD can be present with cirrhosis, MAFLD can be present with hepatitis or autoimmune hepatitis, and so on. So in patients with cirrhosis, we have different strategy because this patient is different from NAFLD without cirrhosis. Patient with cirrhosis, we have to completely abstain the alcohol and look for sarcopenia because the patient is in need for protein supplementation daily, and it's questionable to encourage weight loss or not according to fat mass in patients with liver cirrhosis. Other dietary change for volume management in case of ascites and sodium, potassium and hypokalemia should be looked for. detection and treatment of small intestinal bacterial overgrowth, and the problem of spontaneous bacterial peritonitis because of the intestinal permeability and the probability of translocation of bacteria. So we have to assess also nutritional status with handicap strengths and objective global assessment and anthropometric parameters in these patients. To summarize, reduce the total energy intake for weight loss for long time sustainability, avoid dietary processed sugar, saccharose, or fructose, reduce or abstain from alcohol, incorporate mono or polyunsaturated fatty acids into the diet, consider Mediterranean diet, 2 to 3 cups black coffee per day, avoid snacking, and this is Harvard plate, and this healthy plate for anyone, not only for NAFLD. And I finish with this slide, and thank you.
Thank you, Professor Fouad, for your presentation. Very interesting, also this second session. I have 2 questions, one for any speaker. The first one is for Professor Scarpellini, and is what do you think about the new generation of probiotics to treat NAFLD patients and to for the regulation of the dysbiosis? What is your idea, your opinion on the basis of recent published articles? And the second one is for Professor Fouad. What do you think about vegan diet and its application in patients with NAFLD, in consideration of recent published article that report an improvement of liver steatosis if this type of diet is followed? This last question is suggested by Professor Ivan Rakanovic, unable to be with us today, but my greetings from him and good luck for his life. Please, Emilio.
Yes, what I think, as you also recently said at our Congress of Internal Medicine, is that probably in terms of precision medicine, like also Luigi said in his wonderful presentation, we still need a more accurate fingerprinting of our hepatologic patients, either cirrhotic or non-cirrhotic. So a preliminary fecal microbiota typing would be compulsory before choosing the adequate amount, a type of phyla or probiotic to be administered. Very recently, we see in cirrhotic patients that the control, for example, of Clostridium difficile infection relapse is much more effective if we conjugate high dosage of rifaximin plus multi-strain probiotics administration.
Thank you, Emilio.
Thank you.
Oh, okay. Thank you for your question. I agree with you. This kind of diet is probably a promising kind of diet and management method patient. We don't have a lot of evidence about this kind of diet in our patient, but probably as we know that diet itself or constituents of diet is not as important as the 2 rules: a reduction of calories during the meal, and depending on this effect on the fatty liver. But I think that the reduction of calories in this diet make to weight loss, and this is reflected on the deposition of lipids inside the fat cells and reduction of steatosis. I think it needs a lot of research in this area.
Thank you. I think that this modern diet are a little bit unbalanced because are with low level of vitamins, minerals, fiber, and are focused only to decrease the weight, but in general are unbalanced for health. But
Yes.
this is to the contrary, our diet, Mediterranean diet, we are both Mediterranean, are a little bit
Yes.
healthy.
Okay.
And this is important, I think.
Yes, I agree with you. Yeah.
Thank you. Thank you. Other question? No. Okay, I invite Professor Amadori for the final remarks because he's an important scholar and stakeholder, international stakeholder for liver disease. I invite Professor Amadori for the take-home message of this webinar. Thank you.
Oh, you wanted me to say something about the diet? I can answer with a question to Professor Farhood. What's about the Japanese diet? Why the Japanese are so thin and live so long?
Yeah, they have a lot of fish, and they are very near to Mediterranean diet because they have salmon, they have tuna, they are. I think it's not such diet. It's not only diet. They have a very marked physical activity. They're working so much hard all over the week. I think both the diet control and physical exercise. We can borrow their food pattern as a culture and incorporate this with our Mediterranean diet that we are proud of. That our Mediterranean diet is very important, endorsed, and strong evidence. But we are having the highest prevalence of MAFLD all over the world. So I don't understand this problem.
Actually, Japanese living in Okinawa, they are a little bit different than the other Japanese. Probably because they have McDonald's from the Americans.
Yes. And Kentucky too.
Yeah. So should I say something? Thank you. I have to say thank you to all of you for the really very interesting topic. And and I just enjoy your the rest of the day, and hope to see you, in the future. Diet is continuing to. I mean, when I was a student, I had a young guy who took 6,000 calories per day. He drank 2 liters of Coca-Cola. We didn't talk much about this sweet, with, drinks and Coca-Cola, Fanta at all. You talked about kids, and they probably drink a lot of that. So,
Yes, thank you, Professor Amadori. Finally, in conclusion, NAFLD is the first world liver disease for prevalence. We don't have any recognized worldwide treatment. Of course, diet is important to reduce liver steatosis and to improve our metabolism. And Mediterranean diet is the natural and better regimen, dietetic regimen, to treat NAFLD patients. I think we can conclude all together with these sample messages for our meeting, and I want to my greetings for every of you. Thank you for your support. Thank you to be with us. Thank you to join our conference, and in particular I want to greetings particular, my personal thank you to Hepatoma Research and his staff, editorial staff for this organization. Very good with international speakers, and to Professor Ramadori for the invitation to support us by the journal. Thank you, and I hope in the next future we can organize another webinar with Hepatoma Research support. Thank you.
Okay, thank you. Thank you.
Thank you.
Bye.
Okay,
Bye-bye.
thank you. I think before the meeting ends, let's take a picture together. Can turn our camera, and our assistant editor will take a picture for the memory of the webinar.
Was the best attended meeting with more than 60 people. Congratulations.
Yeah, yeah.
Thank you.
Thank you. Goodbye.
Bye-bye.
Bye.
Thank you.
Okay, thank you.
Bye.
Thank you,
Thank
everyone.
you,
Bye.
everyone.
Bye.
See you.
Ciao, Samir.
Thank you.
Ciao.
Bye.
Bye bye.
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 Bye. Ada, and
Okay.
sì.
Dr. Samir,
Salam alaikum. Yes, sir. Salam alaikum.
Alaikum assalam. Assalam.
Alaikum salam. Alaikum salam.
Alaikum assalam.
Ada and Victoria, after that you can see the certificate, okay? You send us the certificate.
Yeah, yes.
Yes, we will send
You
the
send
certificate.
the certificate. Yes, it's important
Yes.
for marketing and to have a document that we meet to. Okay.
Okay.
Yes, we will send the
They-
certificate to our chair and speakers and participants after the webinar. Yeah.
Thank you for your support. I think is well done, this webinar.
Yeah,
Thank
we
you.
also thank you for your support for organizing this. It's great webinar. Yeah, it's very amazing.
Thank you. Bye.
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 Ciao. Ciao.